Okay, right. So today we are going to discuss about, or we are going to continue the microscope part. Last day we discussed about the basic principles of electron microscope, and now we are moving towards the types of microscope today. First of all, we are starting with what transmission electron microscope. So point number 27. Write down transmission electron microscope. Right. Unit 2, point number 27. Unit 2, point number 27. Transmission electron microscope. In brackets, write down TEM, short form. So first point, write down. This is used. This is used. This is used. This is used to study. To study. To study internal structures of cells. Internal structures of cells. So underline internal structures. Internal structures. Then write down a beam of electrons. A beam of electrons. A beam of electrons pass through a thin. Passed. Passed through a thin. Passed through a thin. Specially prepared. Specially prepared. Specially prepared. Specially prepared slice of material. Slice of material. Then underline beam of electrons there. Beam of electrons, right? Beam of electrons. Beam of electrons, right? Next point, write down. A very thin specimen. A very thin specimen. A very thin specimen is used. A very thin specimen is used. So underline very thin. Very thin. Underline very thin. Write down. Specimen stained. Specimen stained. Specimen stained with heavy metals. With heavy metals. With heavy metals which attach. Which attach. Which attach. Which attach more electrons. Sorry, which attach more to which attach more to certain cellular structures. Which attach more to certain cellular structures than other areas. Than other areas. Then write down. Image reflects. Image reflects. Image reflects the pattern of electrons. The pattern of electrons. The pattern of electrons passed. Passed. Passed through the specimen. Through the specimen. Through the specimen passed through the specimen. Displays on a screen. Displays on a screen. Displays on a screen. Underline displays on a screen, right? Underline displays on a screen. Next point, write down. While electrons pass through the while electrons pass through the specimen pass through the specimen, more electrons may get more electrons may get more electrons may get displayed in regions displayed in regions displayed in regions where structures were where structures were where structures were densely stained densely stained densely stained, right? Pass through the specimen, underline that one. Pass through the specimen. Okay, then point number 28. Point number 28. Scanning electron microscope, right? SIM, right? Write down first point. Here, here means in the scanning electron microscope. Here, a fine beam of electrons. Here, a fine beam of electrons. A fine beam of electrons is reflected. Is reflected. Is reflected from the. Is reflected. Is reflected from the. From the surface of specimen. Surface of specimen. Surface of specimen. Underline reflected. Underline reflected. Underline reflected. Next point. Specimen is mostly coated with gold. The specimen is mostly coated with gold prior to prior to prior to prior to observation prior to observation. Underline gold. Gold. Underline gold. Write down. Here the specimen. Here the specimen. Here the specimen. Here the specimen scatters 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 many electrons many electrons whereas others are absorbed whereas others are absorbed. Underline scatters many electrons. Scatters many electrons. Underline that one. Write down. This is ideal to observe. 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 Surface view, surface view, surface view in three-dimensional appearance. Three-dimensional, it means 3D. Three-dimensional appearance. Underline three-dimensional there, right? Underline three-dimensional. And also underline surface view. And underline surface view. Underline surface view. Okay. So then we'll write down the point number 29. Okay. Unit 2, point number 29, right? Okay. So after discussing that, we'll come to transmission and scanning electron microscope once again, right? Okay. Write down the topic, point number 29, differences between light microscope and electron microscope. Differences between light microscope and electron microscope. So you need to create a table for this, right? You need to create a table for this, right? Okay. You need to create a table for this. Then you can take one side. You can divide your book into two, right? Okay. But don't draw this the dividing line much lengthier, right? Okay. So just go with first start with 5 to 6 lines, then with the right table being right spread, data being entered, you can length it, right? Okay. So you can just draw it more downwards, right? So here light microscope means actually the compound light microscope we are referring, right? Light microscope. Then this side electron microscope. So electron microscope, here we are taking transmission and scanning as one type, right? Okay, transmission and scanning as a one type. First one, write down under light microscope. Okay, under light microscope, first one write down. Okay, side of the light microscope. Glass lenses are used. Glass lenses are used. Glass lenses are used. Glass lenses are used to focus light rays. Glass lenses are used to focus light rays. Glass lenses, right? Then write down powerful magnets in the side of electron microscope. Write down powerful magnets. Powerful magnets are used to focus. Powerful magnets are used to focus. Powerful magnets are used to focus beam of electrons. Beam of electrons. Right? Okay. Then second one. Write down. Image is directly directed by. The image is detected by. Image is directly detected by naked eye. Naked eye. Naked eye. In the side of electron microscope, write down not directed, not directly detected, not directly detected by naked eye, not directly detected by naked eye. Then write down for that one. Micrographs are used. Micrographs are used. Micrographs are used. Not detected directly by naked eye, or not directly detected by naked eye. Then micrographs are used. Right? Okay. Then write down third difference. Living and non-living objects can be observed. 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 In the side of electron microscope, write down only non-living objects can be observed. Only non-living objects can be observed. Only non-living objects can be observed. Right. Then point number 4. And the difference number 4. Write down actual color of the object can be observed. Actual color of the object can be observed in the side of light microscope. Actual color of the object can be observed. Actual color cannot be observed in the side of electron microscope. Actual color cannot be observed. Actual color cannot be observed. Actual color cannot be observed. Then fifth one, dyes used to stain the object. Dyes used to stain the object. In the side of electron microscope, heavy metals are used to stain objects. Heavy metals are used to obtain. Heavy metals are used to stain objects. Right. Right. So light microscope. Now listen carefully. Light microscope is focused or mainly based with visual light. Okay, visual light, the normal visual range that we notice in the electromagnetic spectrum. Okay, electromagnetic spectrum, you know, in chemistry, I think by now you have gone through that one. The spectrum which contain IR, then the visual range from red to violet. Okay, right. Then from there you get ultraviolet, and then X-ray, gamma. Likewise, that the series that is created, the series of electromagnetic waves types which is created on the basis of behalf of wavelengths or frequency wise, right? That order of arranged in order of increasing frequency or in the order of decreasing wavelengths, right? That is called the electromagnetic spectrum. They are that light microscope use the visual range. Okay, visual range. The visual range is used by light microscope. So that's why glass lenses are enough, right? So glass lenses can manage that one. Okay, so those convex glass lenses, they can manage it. But on the other hand, when regarding electron microscope, they use electrons for their purpose, right? They use electrons. Electrons means high energetic kind of radiations, right? That goes beyond the violet range in visual spectrum. Okay, visual spectrum goes beyond violet. It means in ultraviolet, more than that, X-ray, gamma, that region with very low wavelengths, very high energy. Okay, so they go along with electrons, right? very little wavelengths, very low wavelengths, right? Very high energetic, right? Very energetic, okay, with high frequencies. So we can't use glass lenses in handling them because they are highly reactive, and their power and all the aspect with the radioactivity, every aspect of their nature, they are much more powerful when compared with visual light in the visual spectrum. So therefore, powerful magnets are used in the electron microscope. That is much better when compared with glass lenses. Got it? Okay. So then light microscope can be observed by naked eye. We can just put our eyes on the eyepiece that we discussed last week. That we can put our eyes, keep our eyes on the eyepiece, and we can observe with the naked eye. Okay, we can observe with the naked eye. Okay, got it. We can observe with the naked eye very easy. We can directly see the image there, which is formed by the object which is on the stage, no, specimen on the stage. So we can try it with our very fingers, kind of finger on the stage. We can find out that lines on the fingers, that on the palm, that small lines which are somewhat difficult to detect with the naked eye. Then keep it on the stage and we can observe the magnified version. Okay, so we can take it with our real eyes. We can see what is there. Okay, we can see the image. But in the electron microscope, actually, when considering the size of an electron microscope, like compound light microscope, we can carry it with our own hand. We can just carry it with our one-handed carry method. But when regarding electron microscope, it actually occupies a entire lab there, right? Maybe entire room is needed to for the all. It contains a large vacuum, right, etc., and all the powerful magnets, all the powerful rays, all are included there. It's a complex structure, very large, right? Even more, even high, even taller than our size, our stature, right? Okay, got it. So very large machines, very difficult to operate actually. So there, we can't observe it with the naked eye. Even with the naked eye, imagine that X-rays, UV rays, or even gamma rays, or those kind of high energetic radiations coming through our eyes. Then what will happen? Eyes will, you can't even imagine what will happen, right? So in a sudden moment, the eyes will be disappeared, right? Okay. So therefore, in order to protect our eyes and with the difficulty of observing that, that image is projected on a screen, right? So through those micrographs, graphs which are generated by after going through the image, right? The graphs are generated, then those are projected on a screen. Then we can go through that, right? Otherwise, we can't observe with our own naked eye. We can go through that second party option, means that the projected one, like that. For an example, now in your now this online class, you are not getting that experience. But in a physical class, when even we are doing the physical class, we use the projectors, right? When the student count is much more, then those who are in the backbenches, they can't see the board properly. Then what we do, we just place a kind of a screen, and using the projector, we are projecting it, right? Projecting what is writing in the board, and you can see the magnified version. But you are not taking down note with the real one which I have written on the board, right? That's like kind of a thing. Now what I have written, you can't examine it with the naked eye. Those who are in the front rows, they can get it from the naked eye. Others just get the that kind of a projection from the real written note, the board. They get a kind of a projection, then they go through that one, then they are going through the display. Okay, the projection, likewise, that kind of a thing. Then on in light microscope, both living and non-living things can be observed. Even you can keep your maybe anything. You can put a sand particle or a sand, or maybe anything you wish, right? You can put it on the stage. And even living thing, you can put your finger even on the stage. Then normally some organisms are keeping there. Then ants, you can keep them. No matter whether they are living or non-living, we can observe the both, because visual light is going through both living and non-living specimens. But in electron microscope, what happens? We can't actually use living organisms, because that is very harmful. Because it is difficult to manage with what those high energetic rays. That's why living specimens cannot be observed. Actually, they become non-living after that. They are kept under that vacuum, and also specimens are placed in the vacuum. That in the vacuum there is no air, that they can't survive. Actually, living things can't survive. So normally, What happen? Living things turn into non-living things when they are goes through the electron microscope. Got it? So that is what normally happen. So not the living things, non-living things can only be observed with the electron microscope. That living nature or the life cannot be maintained in an electron microscope with the high energetic rays and those complex structures that they are maintained. Then actual color can be seen in light microscope because it uses the visual spectrum for the vision purpose. You can see from red to violet, you can see any color you wish, you can see the actual color. But in the electron microscope, they are not using visual spectrum, they are using something beyond that one, electron rays, then high energetic radiations beyond the visual spectrum. So therefore, their wavelengths are not coming under visual spectrum, therefore they can't see actual colors. So they are going through black and white kind of images there, right? Because they are not having the colors there, right? Okay. Then for the staining process, light microscope use dyes, very simple. With the pigments, with the dyes, they are pigmentation or dyeing them. But for the electron microscope, need to use heavy metals. Can't survive with the dyes, right? With the way that they are doing the complex nature of the mechanism, they have to use heavy metals. Got it? Okay. Then when we come to the visual spectrum, I will go through it, right? When we come to the visual spectrum, okay, so you can get this kind of a rough idea. So we'll do it like this. Now imagine this is the visual sorry, not the visual spectrum, entire electromagnetic spectrum. Okay, got it? So in the electromagnetic spectrum, right? Okay, in the middle you can see what? You can see the visual spectrum there, right? Okay, so this region is the visual spectrum. So this is the visual spectrum, right? Got it? This is the visual spectrum. Okay, so starting from what color? Starting from red, right? Okay, then moving towards what? Purple, right? Purple means violet kind of color. Okay, violet, or maybe violet, we'll take violet. Okay, violet is there. So what happen normally when moving from left to right? What happen? That wavelengths are decreasing and the frequencies they increase, and meanwhile energy also increases, right? This is what happens. So in this side here, you can get here, you can get kind of IR here, infrared, and you can get kind of microwaves here, right? Okay, as different types of electromagnetic waves, right? Okay, electromagnetic waves. Then here after violet, beyond that you can take UV radiations, then you can take X-rays, then you can take gamma, right? What happened by moving towards the gamma, the wavelengths are very little, very small. Wavelengths get decreased. Okay, that's what happened in the normal electromagnetic spectrum, right? So you can see the visual spectrum there. So therefore in this region that light microscope is working. Light microscope is working in this region. That's why they can see actual colors. Okay, they can use living specimens, right? And in this region that electron microscope is working, therefore they can't see actual colors because there is no visual spectrum there, right? Okay, they can't see the natural color with electron microscope. And also they can't we can't observe living specimen under electron microscope. They will get be they will turn into non-living, right? Because of this high energy. Okay, high energy. Got it? So this is the electron microscope range. Got it? Very low wavelengths, here relatively high wavelengths. You can complete it quickly. You can note it down quickly, very quickly. Okay. Now we'll move towards the most important part of the day, right? Okay. So right, so you can now understand. And there also you can write another important fact that we have missed, not missed actually, I intentionally missed that. So there's a reason. You can take a red pen here for this one. You can take a red pen, very important fact. Okay. So with the wavelength, there's a relationship between wavelength and resolution power. Okay. Wavelength and resolution power. Okay. Resolution power and wavelength. Okay. So what is happening? Resolution power is inversely proportional to the wavelength. Resolution power is inversely proportional to the wavelength. Wavelength means the lambda, right? Wavelength means the lambda. Okay, this is inversely proportional to the lambda. Got it? Right? Then what will happen? Right? Okay, when the uh, wavelength is high, right? The resolution power, I will put RP, will be low, right? So this is what exactly happened in light microscope. It means compound light microscope. Light microscope means I will write it compound, right? Okay, compound light microscope. This is what will happen. Okay, then when the lambda is low, resolution power will be very high, right? So this is what happening in electron microscope, right? This is what happened in electron microscope. When the lambda is low, resolution power is high. Okay, so it's a very important relationship. You can use red pen and write it. Quickly write down. Quickly write down. Quickly write down. Okay, relationship between resolution power and the wavelength. Lambda here for the wavelength, right? Lambda here for the wavelength. Okay, right. Okay. So then we'll move towards the most important part of the today's proceedings. That is the difference between scanning electron microscope and right, transmission electron microscope. So first of all, before that, before moving to that one, we need to go through transmission. Transmission. Transmission meaning. Transmission meaning means going through, right? Going through. It means what here, what is going through and which, going through which one, what is going through. Yeah, here the electrons are going through which, through the specimen, right? So the idea in this microscope that electron beams are going through the specimen. Okay, then going through the specimen means going through it. It means able to identify or able to observe, right, internal structures, right? Then scanning electron microscope, scanning, now transmission going through, Scanning electron microscope, what happened? Transmission electron microscope going through. So scanning means scan. Scanning means actually scan. We are scanning what? We are scanning surfaces, right? Surfaces, right? So this is used to observe surface view, no? Surface structures, right? This is used to observe surface structures, surface view. Scan. Scanning electron microscope. Okay, with that understanding, we'll move towards the differences between the TEM and SEM. Okay, uh, we can take we can make a table once again. Here we can use TEM. TEM means transmission electron microscope. Okay, TEM, transmission electron microscope, and then here we can use what? We can use SEM, scanning electron microscope. So first one, the most important one, that in the TEM. Now this is the specimen, right? This is a specimen. It's the specimen, okay, right? So here we use, or the electrons are coming through the specimen, okay. So then here we are able to observe what? Observe the internal structures, ne? Observe the internal structures, right? But in contrast, in the scanning electron microscope, specimen is here. Then what happened? Electrons are coming and they are reflecting, right? They are reflecting, okay. Electrons are coming and they reflect from the surface. They are reflecting from the surface, right? Okay. So scanning electron microscope used to observe the what? Used to observe the surface view. I will color it like this. The surface view. This is used to observe. Scanning electron microscope is used to observe the surface view. The surface view, not the internal structures, right? So first difference is done. First difference is done. This is the first difference between those two. Okay. Then, right? Then the next one. In the transmission electron microscope, electrons are moving through the specimen. Okay. So therefore, we need to use what kind of a specimen? Very thin one. We'll take simple d as the thickness. But for scanning electron microscope, no need of a certain particular thin specimen. We can use a thick specimen even. If you wish, you can use a thin specimen even, right? But the thickness doesn't matter, okay? Because here what it needs, what it requires, electrons need to be piercing through the specimen. In order for that process, thickness is a key factor. Thickness need to be minimized at its lowest, right? Then it is easy. Here what happens, the electrons are reflecting, so therefore no need of having a additional thin nature. Whether the specimen is thick, no issue. Yeah, we can handle it. Okay, that's the second difference. When moving to the third difference here, when moving to the third difference of these two microscopes, third one. Okay, right. What will happen? That here the specimen is what? It is coated with, right? It is coated with what? It is coated with heavy metal, right? It is coated with heavy metal, right? Okay. Then what will happen? Heavy metals, right? Electrons will pierce through, right? Electrons will pierce through, right? Electrons will pierce through like this. Okay. So actually what happened? Heavy metals means heavy. Heavy metals means their mass is high, no? In heavy metals, mass is high. So what happened here? The mg value, that is much more high. Then it is a kind of a additional support for the electrons to pierce in. Then electrons get covered with those heavy metal. Then electrons will be freely goes through, right? In addition to the support given by the thin arrangement of the specimen, along with this heavy metals coated, then electrons will move through the specimen, this thin specimen, okay, because the mg is much higher, right? Mg is much higher. It is a kind of a additional support, right? But in here, what happens here in the scanning electron microscope? So it is coated with gold, right? It is coated with gold actually. So it is coated with gold. Then what happened here? Electrons come and they get reflected, okay? Electrons come and they get reflected. Then furthermore, here comes again, then reflected, comes again, reflected, comes again, reflected. Likewise, electrons will be reflected, okay? Electrons will be reflected from the surface. Okay? Here it used gold. Gold is also kind of a heavy metal, but here the function of using gold is that shiny luster, shiny nature, right? The shiny nature or that glossy nature, okay? That glossy, shiny nature will support the reflection, and further that conductivity is much more, conductivity, the ability to conduct electrons, conductivity is much more high in gold. There are two reasons that gold is shiny, having a shiny glossy appearance, right? Shiny appearance is there now. So that shiny, when a electron beams, even electron, any light, when they get what get attacked on a shiny surface, then they will easily reflected, okay, right? Likewise, so then, and also for the conductivity, gold has a high conductivity, right? Okay. Gold is also heavy metal, but with its different specific characters, it is used for the scanning electron microscope, okay. High conductivity and that glossy shiny nature, okay. Then that is also done for the difference. Final one, right? What happened here? Here the specimen is like this, very thin. Then sorry, right here, then what will happen? 100 electrons, right? And imagine 100 electrons are coming towards the specimen. Then what will happen? Majority are going through. We'll say 90 electrons are going through the specimen. Then what will happen? Remaining 10 electrons will be reflected, right? Okay, this is what happened in uh, transmission. But imagine in the scanning electron microscope, what will happen? They are the same. 100 electron rays comes in, 100 electron rays, and what happened, right? 90 electrons are reflected and some will go through, but that will only encounter 10 electrons will go through the specimen, 90 will be reflected, 90 will be reflected, and only 10 electrons will goes through the specimen. That will happen, that is what that is what will happen there, right? 100 here, 100 comes, 90 the majority goes through, because the aim is to see the internal structures, therefore the majority of electrons need to go through the specimen. Here the aim is to observe the surface view, therefore majority of electrons need to be on the surface. That's why majority of the electrons are reflected and few will goes through, okay, right? That is what happened. Now this is the summary of difference of transmission and scanning electron microscope. I think this is the best summary you can ever get, and this is the best short note. Best short, we have written the note twice, right? We have written the note twice and we have underlined the important facts, but this is the best short note you will ever get, right? For this difference in scanning electron microscope and transmission electron microscope. Quickly hurry up and note this down, okay. Actually comparison is not that much good thing, but once you are actually normally what is you are doing, you are comparing things with friends, no, not only you, even others compares notes. Even when we do A levels, what we do, we compare our notes with friends notes, right? Okay, okay, right. So we can compare, we also in that, in our time we compared and we see, we just we just go through and check whether we are getting the proper knowledge, or in the friends getting more things or more facts. Then we, if there are shortages in our note, we get we just add them in from their notes, and if there is shortage in their note, we'll give them and they will add them into your notes, right? Okay. So but here with the theory batch, the first theory batch we are doing, Actually, I'm giving everything possible, right? Everything needed, actually, not going through the unwanted rubbish, right? I'm not filling your notes with unwanted things. I'm going through the all the necessary things, and they are provided with easy kind of manner, smart manner, right? Very easy to understand. You can see. So then you will be very thorough with the theory, right? Okay. Right. So that is the difference between transmission electron microscope and the scanning electron microscope. Very simple, right? Very simple when you are going through. So those are explained in the note, and I just kind of given you a kind of rough sketch idea. It's actually a very interesting short note, right? You can have all the necessary points needed for the electron microscope in a nutshell, right? Okay, got it. So all are included there. All are included in a nutshell, right? That is the importance here. Got it, right? So right. Then today we had that electron microscope two types of electron microscope to discuss: scanning and transmission. So transmission electron microscope, when you go through this side, you can see that always the electrons are pierced, electrons are going through. That's why the name itself suggests transmission. So majority of electrons also going through the specimen. In order for that, there are several adaptations that the specimen is prepared in very thin arrangement, and also coated with heavy metal in order for the supporting of electrons moving through very quickly, that the mass of the metals will also support. Then same scanning means the surface view, the name suggests scanning, it scans the surfaces of cells in order to get the 3D structure of the surface view. Then the thickness doesn't matter, because even thick specimens no matter, because we are only going through this surface. So there are several and also the majority of electrons are reflected, not going through, and several things we are doing in order for the proper arrangement of the specimen, that we are coating it with gold. Apart from heavy metals, gold is also kind of a heavy metal, but apart from that we are using special heavy metal gold, because gold is kind of a shiny metal. It provides some sort of luster there and glossy nature. So then that will help for what, the reflection, for much more reflection, right? And conductivity will also be increased when compared with the other metals, right? If you use, right, so conductivity also increased, then that will favors its reflection, but it needs, right? So these are the differences in scanning and transmission electron microscope, and we discussed the difference between light and electron microscope. And one thing, very important thing, if you ask to write down similarities between transmission electron microscope and scanning electron microscope, what will you write? You have to write, right, that factors under electron microscope, in that table, differences between the electron microscope and light microscope, the factors listed under electron microscope actually are the similarities between transmission electron microscope and scanning electron microscope. So the points listed under the electron microscope actually the same for both transmission and scanning electron microscope. Heavy metals are used, okay. Then electron beams are used. Then electron magnets are used, okay. So actual color cannot be observed, okay. Only non-living things can be observed. All are common for both TEM and SEM. That's it, right? So that's what we have to do with today's proceeding. So then the next day, coming day, we are moving towards the most important part of the unit 2, the cell aspect. Cell aspect. Prior to that, we go through the microscope, okay, right? Cell aspect. Then, and also one point we discuss about that resolution power and the wavelength relationship. Keep that part in the mind also, right? So, and cell basis, we'll start with the cell theory. These are frequent, most very common things for you, because you have done those things in the O levels too. Cell theory, then cellulose, but we are going through much more deeper than what you have done in O levels, right? Okay. So then now we are piling up theories, so it's better to go through the theory parts and study them well, right? Otherwise, there will be kind of, the things will be piled up. Then it will be difficult for you to catch up once the exam is there, right? When the exam is much closer, right? When the exam get closer, stress increases, right? Stress rises up, then that will be difficult for you to just arrange things, right? Not like O levels, right? A massive content, right? You can see now it's still early days, but you have written much more lengthier notes, right? Much more content, much more facts, logical parts. So think and study well, and also not only the biology, rest of the subjects too, right? So all the necessary things, it's up to you. The preparations, the dedication, determination, it is up to you. Not that we can provide all the necessary things. 50% will be from our side, and the rest of the 50% you need to be completed with your aid, right? Otherwise it will be difficult, okay. So then till the next day, next week, goodbye, okay. So enjoy the week, right? Okay, coming week, and keep doing on the studies, right? Okay, likewise, then the O level results will also be released sooner, then you can enjoy more, right? Okay. Share your happiness also with us, with us too, right? Then we are ready for your happiness. Happy news that so I have got 9 years. Okay, nice. Then it's the next mission, you need to get 3 years. Really possible if you work hard. Okay, so that's up to you. Okay, till then, goodbye, have a nice day, good night. Okay, we'll meet on next Sunday with the sale, right? Okay.